All right. So thank you, everybody. I don't have too much to say before Omar gets started, so we'll get everybody off to lunch here in just a minute. But do want to go over just a couple details, remind people of the links for the hackathon materials. Just as a reminder, this session, even though it's short, is still being recorded and live streamed to YouTube, just so everybody is aware of that. So this information is more or less the same as yesterday, and everybody should have gotten a long email from me last night about all of these details for getting your information ready for the hackathon. So I'm not going to go over the schedule in super detail today. Again, this is in your email inbox, and we can send it out again tomorrow in our kind of final update email about that. So we will be starting at 10 a.m. on Friday, August 9th for team organization. Just a couple of things about preparing for hackathon teaming. Again, if you have not filled out the hackathon interest survey, please do take a minute to do that. And I'll drop these links in the chat while Omar gets set up in a second. Again, please do add a brief idea or a brief description of your idea to the shared Google Doc if you have a project idea already. And if you don't, please do put your information in about what kind of group maybe you're looking to join, what kind of work you would like to do. And we have our project ideas document linked here as well, if you would like to take a look through that over the next couple of days.
Hello, my name is Omar. I'm a PhD student on applied physics, and my project's idea is to develop a rat tails phantom for irradiation. So the idea is that on radiotherapy, the radiation delivery rate is an important parameter that determines the biological damage. For example, it's been shown that ultra high rates spare normal cells while maintaining its effect on malignant tissue. This is known as the flash effect. So, for example, the number of particles that arrive at a certain time interval is the dose rate. So, the is not about the total dose, but like how many particles can arrive to the cells at this time interval. So another factor that influenced the effectiveness of this flash effect is the level of oxygen in the tissue. It's been proposed that the flash effect is related with the depletion of oxygen on the cell's environment. So we already have used CC3D in combination with another software that's called Topaz, that it's based on Giant4, which is a Monte Carlo track structure code to simulate the radiation transport on matter. To simulate the cell irradiation, we are we have an ongoing publication, so it's already on the editors and so on, of an in vitro configuration. This is a monolayer of a cell of PC3 cells irradiated with X-rays. However, the goal is to simulate more complex tissue models and its response to radiation, including a realistic distribution of oxygen. So, for example, CompuCell generates the cell distribution. The geometrical information is passed to Topaz, which simulate the irradiation. Dose is calculated, and the biological process continuing is feedback to CompuCell and is continued. So there are a lot of experimental data on rodent models, especially on the effects of radiation rates on the biological effects on rat tails, which are necrosis, skin damage, fibrosis, etc. And there is also abundant information of the physiology of the rat tails. So the idea is to develop a realistic section, like about a millimeter cube of the rat tail, with the main artery explicitly simulated to generate the oxygen distribution. The objective of this model is also to change the artery flux to simulate different oxygenation conditions. Like in the experiment, it's done with a tourniquet, so maybe changing like the what's it called, like the oxygen flux. I'm sorry. So we have already a 2D prototype working on each XC and YC planes. So the artery is this red dot. This is the muscle, and these three layers are the epithelium. The red dot have a constant production of oxygen, and we have the constant oxygen concentration of this value, and these are the values that we are taking from this publication from Shirin. I hope I'm pronouncing that right. But we want to extend it to a 3D model, but we have like several issues to solve. For example, cells have different sizes and oxygen consumption. For example, muscles are longer fibers that consume much more oxygen. We need to know if this is like the correct scale, like this is a ton of cells being simulated. So we want to check if the voxel to volume equivalence and Monte Carlo steps to time equivalence are correct, or if we need more detail and focus on a smaller area. So other issues that we are using values from another model, but we don't know if for example, the oxygen consumption on rats is different. So we want to extract them from experimental data. And for example, it's just the artery, so this whole system equilibrates slightly slow, but we want to maybe add other capillary structures like these branching veins, but we don't know if that's realistic. And for example, one thing that one of the biological effects are the fibrosis. So maybe besides the DNA damage, maybe we can even simulate the chemotactic response of fibroblasts that are recruited to the damaged sites, because that's an effect that we have been trying to simulate. So we have some literature. I have even more if people are interested. And I am, as I said, a PhD student on applied physics. I know, not an expert, but know medical physics, Monte Carlo radiation transport simulation, specifically on the code Topaz, now Open Topaz, and Python and some C++ programming. I would, if someone is interested, like I really don't know anything about biology or biochemistry, but like I just take the models that we research and apply it. But to develop new models, like I'm not really sure. But well, that's it. Thank you a lot.
I think that looks promising. I hope maybe there are other people interested in this radiation coupling. I put a couple of papers that have looked at it either with CompuCell or other packages in the chat as possible things to think about, but you've already got a lot going on. So